Thank you.
안녕하세요. 주한 이스라엘 대사관 무역투자대표부 신종혁 상무관입니다. 오늘 바쁘신 가운데 참석해 주신 모든 분들에게 진심으로 감사드립니다. 오늘 행사는 영어로 진행될 예정이며 지금부터 영어로 진행하겠습니다. Good afternoon, everyone in Korea. Good morning to those joining us from Israel. Thank you for joining us today. My name is Jong-hyuk Shin, and I'm a trade officer covering the healthcare and cosmetic sector at TradeIL Korea. And I'll be your moderator for today's webinar. Today, we'll hear from some of Israel's leading pharmaceutical and healthcare companies and learn how they are using AI to accelerate drug discovery, improve diagnostics, and drive innovation across the healthcare industry. Before we get started, just a quick housekeeping notes. This webinar is being recorded, and also, if you have any questions, feel free to type them into the Zoom Q&A box, and we'll do our best to address them. Now, it's my pleasure to invite Tamar Kosher Maron, Head of TradeIL Korea, to deliver the welcome remarks. Tamar, the floor is yours.
Hello, everyone, and Yasel and Shalom. Thank you so much for joining us to this special occasion. It is our pleasure to have with us Amir, Eran, Mati, and Tal, and showcase amazing ideas and innovation coming from Israel. Thank you for joining from across Asia, and I hope you will enjoy today. For any question you have, you can just type in the Q&A box. And if anybody wants to meet these lovely fellows, just let us know, shoot us an email or DM us, and we will be happy to set an appointment for you guys. Thank you, and all the best.
Thank you very much, Tamar, for the well welcome. Now, let's move on to our keynote presentation. I'm delighted to introduce Mati Gill, CEO of Aion Labs, who will be speaking on how AI is shaping our healthcare and future pharma landscape. Mati, over to you.
Thank you. And it's really a pleasure to be here. And thank you, Tamar, Jung, and all the embassy team. And it's really fantastic. And you have much smarter people after me that will give you great case studies from great Israeli companies. So I'll try not to embarrass us in the meantime. And I'm going to just share a little bit about where Israel is continuing to be the place, as we have emerged over the last 5-7 years, to be the place, or one at least of the places where AI meets biology. I must say on a personal note that I had my first opportunity to visit in Korea last fall, and I've been to some of the countries that many of you are also situated in, but I was very, very much impressed by the level of technology in Korea and hope that this is just an opportunity that we can start to build bridges between Israeli companies, and including Aion Labs that I have the opportunity and honor to lead, as well as with companies in your nations as well. And we even have one example where we're actually doing that actively. So Aion Labs is a venture studio, and we are backed by great leading pharma companies, including AstraZeneca, Merck, Pfizer, and Teva, that came together to build up a venture studio, to build startups in Israel, working as competitors, together with good venture capital funds and supported by the Israeli government as a public-private partnership. And what we're going to talk about today is a little bit about AI and technology in drug discovery and development, why Israel is a good place for that, and then I'll give you a couple examples with regards to what we do at Aion Labs. So first of all, just like any startup, and just like Scopio and Protai that you're going to hear from directly, we at Aion Labs are trying to solve a problem. And we are part of solving a problem in this case. And this is where the opportunity lies for technology and for artificial intelligence and machine learning and other computational drugs, is a problem statement that you see right here, is that it takes way too long, way too expensive, with way too high a failure rate to bring a new drug to the market, because we are basically, as an industry, continuing to discover and develop new drugs the same way we've been doing it for 100 years, trying it out in labs, trying it out in animals, and then taking it through clinical trials in patients through multiple different cycles. So well over $2.5 billion on average, over 10 years, closer to 15. But I think the most striking figure here is the one where we fail over 90% of the times. And that is what one of the biggest drivers in why it's so expensive and why it takes so long. Because we fail over 90% of the times once we actually start to test out a drug in patients, meaning that we are not being efficient enough as an industry, and we know that this is unsustainable going forward. Now, that being said, there's an opportunity, and the opportunity right now, and why this is such an exciting industry to be in right now, is because biotech is becoming an increasingly technology and data-driven industry. So biotech is made up of two parts, the bio and the tech. And as you're going to hear from some of the examples you hear from today, technology is becoming an increasingly important part of that biotech equation, between the capability to actually learn from data with large machine learning models, artificial intelligence, large language models, automated processes, and connecting engineering with biology, two very different scientific disciplines. It's much more of an opportunity actually to enter into this space if you're coming from an engineering and technology background. And this is going to be the future of our industry, is going to be much more technology driven, not getting rid of the biology, not getting rid of the life sciences and chemistry, but complementing it and helping to unleash it by unleashing the full capabilities of technology to help enhance it and enhance our learnings there. So why Israel? Why we are actually a good place? Number one is and you'll see some of these examples, and I'll share with you some of them. We have seen an emergence over the last 6-7 years of a new generation of biotech companies that are AI and life sciences driven. We have a great entrepreneurial culture. So basically, if we have good life sciences capabilities and have good technology, we also know how to build products, connecting those two scientific disciplines, and then help build startups in that space. And there's a lot of a culture that enables that. The science in Israel, and I think this is the real foundation, the science in Israel is world class. So it's really on par with some of the best scientific capabilities that you'll see anywhere in the world. I'm speaking to you here from our site in Aion Labs. We're right across the street from the renowned Weizmann Institute, which is one of the top 5-6 scientific institutions in the world. And we have 6 or 7 other top world-class universities with great labs that are spinning out every single year great technologies and science. Our ecosystem also works very much together, and we help each other, and it's a very good culture of trying to propel the ecosystem at a national level. And the technology capabilities of Israel, I think, are very well renowned, and that is true in software, and it's true also in deep technologies. These capabilities exist, and we are seeing more and more global pharma take notice of Israel in that sense. And I think this is also very much the same in Korea. The government of Israel is very much behind and supporting of the growth of the biotech industry. And so just like I mentioned before, we have seen an emergence of a whole cluster of Israeli biotech companies. Most of these are technology driven and really at the forefront of connecting technology and biology. And I would say almost all of these have been built, if not all, have been built over the last 5-7 years. So almost none of these companies existed a decade ago. And so this is what we are seeing, and this is just a few. I could share even more if I wanted to. But all of these companies have pretty much emerged over the last 5-7 years and have started to grow. Some will succeed tremendously, some less, just like in the nature of any startup. But Israel has a promising cluster of great science-driven, technology-driven biotech companies that are emerging here. And it's something that makes us very optimistic about the future of the ecosystem in Israel. And all of these are looking to work globally. And as I mentioned before, we are seeing increased interest. So this is just a snapshot from the last decade. And you could see as the decade goes along, you're seeing more and more interest of global pharma companies to work with and to work in Israel and to work with Israeli companies, including those like GSK that have started to build up local R&D centers based on AI and machine learning, and global pharma companies that are doing collaborations, sometimes at hundreds of millions of dollars worth of value with Israeli companies, and this is just increasing as we go on. And part of this is because we are seeing globally, not just in Israel, obviously, that we are seeing new technology players enter into this space, and it's bringing in new resources, new value, new capabilities. Eli Lilly has been at the forefront of this, signing strategic deals both with venture capital firms, but also with NVIDIA, and enabling startups to access their data through new technologies that were built with Israeli companies like Aion Labs, that was built with Rhino. Amazon just last week, Anthropic released their new cloud for sciences. And we're seeing more and more of these technology players enter into our space, understanding the potential. And this is just the beginning, and including making acquisitions because they want to bring in the capabilities and the expertise. So this offers new exit pathways for startups that are building technologies. It offers new resources and also new players that are bringing in tremendous capabilities into the space in the compute area, but also that can help the startups grow. So I'm going to pause and move over to Aion Labs and how we are taking advantage of this opportunity to help build up the ecosystem in Israel and to help build up startups. And really, we are doing that in Israel by connecting all of these partners that you see here on the slide, 4 pharma companies working hand in hand as equal partners, Merck and Teva, alongside Pfizer and AstraZeneca, that are 2 of the top 10, maybe even top 5, depending on how you count, pharma companies in the world, all working with their global research and development teams in order to bring in those capabilities to work with startups in Israel at a very early stage. Amazon is a cloud and technology partner, venture capital funds with the Israeli government funding to help de-risk everything, and then reaching out to talent from anywhere they come in the world, working with ecosystem partners to bring in the best talent from Israeli universities or globally. We have several entrepreneurs that come from abroad, build startups with us, and work in a very hybrid manner in that sense. And over the last 4 years of company building, we have built up 9 startups, worked with a 10th in an open innovation model, and really started to see the way that artificial intelligence can help us to develop new technologies, to develop insights into the way we understand the disease mechanisms so that we can have new understandings of the way human disease biology works in the brain, works in different disease mechanisms in the makeup of human disease biology, and then to be able to discover new drugs, both in small molecules, large molecules, as well as in other new modalities. And then starting to work in areas that are very tricky, where there's still a lot of opportunity in preclinical development to replace very non-predictive models of how we predict which drug will behave well in the clinic, in clinical trials with patients, and then to be able to connect good companies that are able to help navigate clinical development with our global pharma companies, like we did with Phase V, that is really at the forefront of helping to navigate clinical trials successfully and to reduce that attrition rate from 90% to something much less. When we build a startup, we ask 3 fundamental questions. We could start with the need, or we could start with the team and technology, or we could start just with a person that we think is incredible and then help them to build a good startup. But always they have to have 3 core principles at a startup when we build them. Number 1 is that they are in the right space, meaning that they're addressing a fundamental big problem statement that we very thoroughly define. Number 2 is that it's a great team and technology, and the team also has to have capabilities both on the life sciences as well as on the tech side. And number 3 is that we're the right place, meaning that we have at least 1, if not more, of our 4 pharma companies that are willing to do early proof of concept studies with the startup to bring them to a point of validation and to help them succeed, to validate their technology within the first 2 years so that they can define a product, raise their next rounds, and continue to graduate within 2 to 3 years. And or if they're not working precisely, if the technology is not working well enough, then we can shut them down. And that's what a venture studio does and move on to the next thing. You try something out, doesn't work, you move on. But I think this slide captures what's at the heart of our collaborative and co-creation model is we have actively with us now 7 startups, including Combinable that we sold to In Sitro. And all of these 7 startups are doing various proof of concept studies or have them planned with different and have them approved in theory with different pharma companies from within our partnership, and also helping them because our pharma partners do not take intellectual property rights. They also help them. We help them to reach out to other global pharma companies and reach proof of concept studies with them as well. And each one is able to actually validate their technology, whether or not it's actually working. If it works, great. If it doesn't, then again, like I said, we can move on. And what's unique about our model is that when a startup starts out with us, they have direct access to some of the best R&D experts in the world in their specific field. So if you're in the protein engineering field, like Denova is, then they have direct access to the heads of biologics engineering of AstraZeneca, Pfizer, experts from Merck and Teva, et cetera, et cetera. And for each one of these startups, it's a different set of experts based upon their specific R&D area. So just to share 1 or 2 examples, we came with an opportunity and a need that we called multi-objective optimization of antibodies, being able to actually build a machine learning-based capabilities. And we came with this as a challenge, an open challenge. Daria, who you can see her picture on the top right, she's a computational biologist, a scientist that came from Germany, and she wanted to work with a great Israeli AI machine learning expert, someone who actually, and we found Noam, who actually wrote a book on artificial intelligence after doing his PhD in Beersheba in the machine learning and engineering field. And they built up this startup, this technology that was able to actually win global contests of Ginkgo and global head-to-head contests of that In Sitro, one of the best biotech machine learning-based companies in the world, was able to do. They had great results that were able to beat the whole field in this sense within 2 years, with $1 million invested in them. And ultimately, after 2 years, they were acquired by In Sitro, that has now established them as their R&D site here in Israel. And the first time a US biotech company has actually established any R&D presence in Israel. And hopefully that will just grow based upon that, because one of the things that they're looking in is to tap into Israeli artificial intelligence talent here in Israel to be able to bring them into the biotech space. The last company I'll share with you here is a company called Profit. They're in the small molecule design space. 2 friends that had known each other for a year, with a third co-founder that comes with a computational chemistry background. Basically, what they were saying is they want to be able to design and find the right small molecule that can target any disease out there without starting with an initial structure. This is something that the pharma company said was a huge unmet need. They've already been able to show within under 2 years, actually, they haven't been around for 2 years yet. They've been able to show within 2 years that they're able to outrank the competitors. But in addition to that, they've actually been able to both outperform their competitors, but able to actually provide value to different pharma companies out there, including a great company called Dongbang from Korea, that together, Dongbang is actually developing their own molecule based upon machine learning technologies from Profit. They jointly applied to a funding opportunity from the joint Israel and Korean fund called CORIL, and able to actually receive government grant to reach out. You can be in touch with us. You can help us connect with partners locally. If any of you are pharma or biotech companies or know of biotech companies that are looking to tap into AI and machine learning capabilities, please reach out to us. If you want to invest in good growing companies, we have a lot of opportunities to do that, or to invest in funds. We're happy to help you as well in that sense. And if any of you are very talented people that want to work in this field, we're open for that as well. And I'm going to leave you here with all of my details. If you would like to reach out to me personally, it's easy very much on LinkedIn, Mati Gill, or you can actually reach out to me on my email, which I'll put here in the chat. But it's Mati, M-A-T-I, at ionlabs.com. It's very easy in that sense. And Tamar and the embassy team can give it out freely as well. We're very accessible. Thank you. And I'm going to hand it over to people that are much smarter than me now.
Thank you very much, Mati, for the insightful presentation. Now we'll move on to our company presentations. Our first speaker is Tal Almog, Senior Account Program Manager at CytoReason. He'll be sharing how CytoReason is using AI-powered disease models to help make smarter clinical development decisions. Tal, the floor is yours.
Thank you very much. Again, I'd like to also thank for giving us the opportunity to present CytoReason today. It's a great pleasure, and I think that as Matti mentioned, there are multiple Israeli companies in the field of using AI models and using deep tech for pharma development, and CytoReason is actually one of them. So I will go directly to what CytoReason does and how we work with pharma companies and biotech companies. And basically, maybe just a few words about me. I'm working with customers directly. I'm leading the part of customer engagement at CytoReason. I can tell you it's definitely an interesting place to be at and exciting. And if there are any questions during the presentation, please feel free to put them in the chat. So CytoReason is actually working with our partners and using AI models to understand disease biology and drive R&D portfolio decisions. And I won't go on the data that actually Matti previously shared, but you can see it's consistent. It is one of the challenging aspects to develop a drug. And we say it's not because the science is not rigorous that drugs fail. It's because the diseases are extremely complex, and disease biology is also very complex, which basically changes between diseases, patients, tissues, and over time to make it even more difficult to develop a drug. And this is actually where we say, there is a lot of data out there, especially through the last decades, multiple data types that include clinical data, omics data, transcriptomics, proteomics, you name it. Now, the question is how to take all of this data into one coherent picture. And we say that basically we need an intelligence layer to solve some of the key use cases that CytoReason is working on. Oops, sorry. So if you look on the right-hand side, these are the four use cases that CytoReason is engaged with, which is, first of all, to understand the disease biology, to better develop the target that we are interested in. Second of all is to differentiate the target versus the standard of care and the competitions, which is key before anyone invests money in developing a drug. Combination strategies, whether our target would work well with other targets or not, and in which diseases. And lastly, which is key questions for pharma companies that are entering a crowded market, which patients populations are relevant for our drug. So that ultimately we get an improved understanding and that leverage better targets and smarter trials. And this, we believe, can help and change the way that drugs are being developed and make the process more efficient and more successful. So how do we actually do that? So here you can see a very high-level picture of what AI disease models at CytoReason are. Okay, we use a structured way to understand biology. So basically, we take data, various types of data, both molecular data, clinical data. It can be proprietary or customer data. And then we add another layer of knowledge that includes language models, KOL opinions, and node biology. And lastly, we add the layer of machine learning that takes multiple data points and data stats and incorporates all of that into one coherent picture, which is the CytoReason disease model. What can we actually do with the model? So first of all, again, I must say our models are relying solely on human tissue. We were not using any mice data, any synthetic data, or things like that. And so the models can actually be very close to what a disease looks like from the biology angle. So we can look closely into cell composition, regulatory programs, networks in the tissue, the progression of the disease, which is again a very important aspect. How does the disease look like after a patient has been treated, as well as the heterogeneity of patients, which again can be the difference between success and failure in a clinical trial. So if we are focusing again at the use cases that are of interest for pharma companies. So before going into a target development, we would like to prioritize targets, especially in very early stage R&D. There is constant prioritization of targets. So we want to make sure that the right target is backed before we commit long years of R&D. This is what CytoReason does. We want to make sure that the combinations that we're choosing are actually working together. It might be the same mechanism, like doing a double hit. It might be different mechanisms. It's just the matter of the selection of the customer sometimes. But it's very important to know what are the options. Going to additional use cases, so we have indication prioritization, or some pharma companies might call this life cycle management. Which diseases are we interested to enter? And what's the rationale behind it? If we are currently at preclinical stage, we want to prioritize from multiple diseases, and this can be, again, the difference between a successful or a failed drug. So we want to make sure that we have the right rationale to enter into one disease or another. This is disease prioritization, and we have patient subpopulations, which again relates to patients that might respond to the treatment. And this is very important when developing, especially second, third line of treatments, that we want to look at non-responders to one drug and predict which patients of the non-responders might respond to this drug, to the drug that we are developing, right? So these are key questions that we are constantly working on, and I believe that focusing on the biology can actually benefit the development. So a little bit of how partnership with pharma company looks like typically. CytoReason has a platform. We have a graphical user interface, and we have the computational environment over which the scientists of our customers can actually work and explore and reach their own conclusions on a self-paced way. In addition to that, to empower and to add other layers of sometimes more complex use cases, we provide a layer of scientific services. And this layer enables working with our scientists using the CytoReason disease model and the platform. And the third layer or pillar, you might look at it as a pillar, is collaboration analytics based on customers' needs. We develop novel frameworks for insights. So taking all of that together, we provide a comprehensive solution for our customers. So how does the platform actually changes the way scientists look at the problem of developing drugs, right? So the old question used to be, does the drug work at all? And using the four use cases that are presented above here, we can look at which targets are we developing the drug for, which combinations, which indications, and which patients might respond to the drug that we are developing. So the questions that we are asking are more focused and more directed into the new and the more data-driven world that we are living at, and focused and direct to these questions. I'd like to present to you also Lina very briefly. Lina is CytoReason AI agent. It is, similar to other agents in the sense that the user can use free text to get insights. This includes plots, it includes code, and includes insights like summaries. The only difference between Lina and other agents that Lina is directly connected to CytoReason disease models. So the user can actually benefit from the models in a very friendly way, and that actually gets them right to the point they want to be, and they're not tied into specific interfaces that are more fixed, and actually provides the answers to the questions much more quickly and friendlier way. So CytoReason is developing these tools not to replace the scientific judgment. It's basically to give the scientists a deeper understanding of the biology, the complex biology that they're reasoning for. And this is what we're here for. I'd like to conclude and say that CytoReason has been around for 10 years nearly, been working with some of the largest pharma companies globally. Our platform has been deployed with some of the largest pharma companies. It's been validated by them, including internal data and publications, joint publications, and as well as scientific papers. Has been using at scale with our customers, and I'd like to just add that again, we're witnessing a very interesting era where the biology is not like traditional science anymore, and we're looking at more like an engineering problem. This, of course, takes time, but I think it's progressing in the right way, and we are happy to be part of it. So. Again, I'd like to thank you all for inviting us and the opportunity to present here. You're welcome to reach out to me and via this email tal.almog@cytoreason.com or through our embassy, the delegation in South Korea. So thanks for listening, and do let us know if you have any questions or thoughts.
Thank you, Tal, for that great presentation. Next, I'd like to welcome Aaron Seger, CEO and co-founder of Protai. Protai combines AI and proteomics to discover new drug targets and biomarkers, helping speed up the development of next generation therapies. Aaron, over to you, please.
Hi, everyone. Very nice to be here, and thank you for arranging this amazing delegation or webinar. My name is Eran Seger. I'm a CEO and co-founder of a company called Protai. We are focusing on proteomics and AI for drug discovery. as you all know, a very hot space at the moment, and Israel, as Mati showed us all, is becoming a significant hub in this combination between AI and drug discovery. Here I'm on the bottom, you can see my email, so feel free to reach out. I'm approachable. So as Protai, we're focusing on the unmet need of induced proximity. Induced proximity is an emerging space of drug discovery, and we are opening the induced proximity space for structural proteomics, using structural proteomics and AI for rational design and structural-based design, where traditionally these drugs were designed by trial and error methods. This opens up modalities such as Protax, molecular glues, and Riptax. We're actually in molecular glues collaborating with Standime, a Korean company, also through the Korea-Israel collaboration grant. So, and I think Sangok is also here on the webinar. So, also a significant hub in Korea. You can say that we're basically couples therapy for proteins, and we're helping them stick together. We need 2 or more proteins, and we're gluing them together in an artificial way using the power of AI and structural proteomics. Why is this hard to do with traditional methods? So 2 main traditional methods to map ternary complexes and complexes as a whole. One are crystal-based methods like X-ray and cryo-EM. These methods are on one side well established, but on the other side are very slow and static. And as a static freezing of the protein, they don't really mimic what's really happening in the biology, and they have their limitations. And some companies are trying to overcome this with AI approaches, but when it comes, while AI is very good on a single protein and the binder, binding affinity, when it comes to 2 or more protein, it starts to making things up, and the structural AI tools are just not good enough. And in order to effectively design these drugs, you have to really model what's happening in the protein dynamics and not only in a static state. And in order to do that, we're adding a layer of data from proteomics. To overcome these liabilities, we've generated what we call the AIMS platform. AIMS stands for AI and MS. MS is our tool for proteomics, mass spectrometry. And this enables us to create a dynamic and functional mapping of the protein complex, and enables us to design best-in-class drugs for these protein complexes. This platform has 2 parts. The first part is AIMS-Fold. It's our take on AlphaFold, which takes into account also experimental structural constraints. We are generating proprietary data in our mass spec lab on 2 kinds of data. One is data on the surface exposure, so how the proteins are touching each other on the surface using a method called HDXMS, and I'll not go into exact technical details, but you can approach me afterwards and I can send you some additional materials. And the second constraint is the distances between the proteins using covalent crosslinks between the proteins. And these together give us experimental ground truth to what's really happening in the native state of the proteins when they touch each other. This is embedded as part of the AI model in order to create what we call experimentally grounded AI model or proteomic-enabled AI model to generate the best-in-class or the state-of-the-art model for the ternary complex, and enables us to surpass all the conventional models such as AlphaFold and Boltz. Once we have the state-of-the-art model, we can use this to rank the best molecules, and here we're using AI affinity on the dynamic model in order to prioritize the best compounds and synthesize them and so on and so forth. This is also a very rapid method, so it takes us less than a week to generate all this data per compound, and it enables us twofold. One, it's very relevant for the timelines of drug discovery, the DMTA process, the drug design, make, test, and analyze the cycle, which if you look at the crystal structure, it takes a few months. Here, it's less than a week, so each batch of compounds we can test in the same method and generate high throughput and large amounts of data. And the second is that because this is a very efficient method, we can generate a lot of models per program. So this is a target-centric approach where we're generating more than 30 to 40 models per project on the best compounds that we have. And both of these are very intriguing when it comes to drug discovery. Just to emphasize how this works, so here in the middle, we have a ternary complex of two very much well-established proteins, WDR and DCAF1. It's the E3 ligase and a target protein. And you can see that in crystal structure, there's a significant difference between active and inactive compounds. So on the active compounds, you can see that this black smudges on the bottom side, when in the inactive, it's on the upper side. When you do AI naive prediction using a method called Boltz-2, you can appreciate that there's no difference between the inactive and active. So it gives more weight to how the proteins are folding on themselves and doesn't give enough weight to the molecule itself. And in order to overcome this, we're adding constraints from covalent crosslinks, and this enables us to create an Ames fold model, which was 99% correct and maintains the differences between the AI naive prediction and the Ames fold, and the active and inactive conformation like the crystal structure did. And we just published this in NeuroIPS preprint, and I'll send it also afterwards on the chat. We also benchmarked this model, our model, and 6 other use cases in a target approach manner. So it's a very promising method. The next generation of Ames will be, so everything I showed until now was using recombinant proteins. Recombinant proteins are proteins that we're expressing artificially, which is very common in drug discovery, but it doesn't really mimic the native state of the biology. And in order to mimic what's really happening, we're moving these methods from expression system to cellular systems. And this enables us native biological analysis of what's really happening in the cell. We're using methods of crosslinks and pull down, so we're using still a target-centric approach, but here we're maintaining the post-translational modification space and the full structure of the protein in its biological state in a cell-specific manner. This enables us to generate a much more accurate modeling of what's really happening on the biology side. And this will be available also in a few weeks. All of this is not only an engineering drill, we also are generating, our main business model is to generate our own enabled or Ames-enabled drug pipeline, and we have several drugs now in the pipeline. Our first drug is an oncology drug, degrader for a target called KAT6A, which we're taking to clinic early next year. And on top of that, we have several assets that are in the autoimmune space that are now in discovery and preclinical stages. We also partner up with biotech and pharma companies. So we have 2 models, and I'll speak about this in a sec, but we have one already partnered asset with a biotech company in the areas of aging. So now to explain how this works in a real life scenario, I'll show a bit about what we did with the KAT6A degradation. And here, apologies if this is too much depth in science, but we're a scientific company, so I think it's important to show how this really helps in discovering a drug. Here we took a complex and a target called KAT6A. It's a highly established target developed by Pfizer in a phase 3 clinical trial. It's an epigenetic regulator of histones that regulate hormone-based proteins and oncogenes, and are a key factor in oncology and beyond oncology. The problem with KAT6A is that it's very much preserved between the family members of KAT6, KAT6A, KAT6B, KAT7, and this didn't enable to generate high selectivity, as well as the fact that it works in a complex which can be compensated by other complex members, and both of these shoved us towards generating a Protai approach or a degrader approach that gave us a high selectivity because of the differences in the ternary complex, and B also overcame the scaffolding activity of KAT6A as a protein member and generated much stronger activity compared to the enzymatic activity that Pfizer has showed, and enabled us to generate a first in modality, best in class drug for KAT6A. This started with generating a ternary complex model using our Ames fold model, and here we successfully used HDXMS and XLMS together with the AI modeling in order to distinguish between unproductive and productive conformations in the ternary complex. And we found out that while there's one angle that is unproductive, other products generate highly productive compounds with high cooperativity between the E3 ligase and the target protein. And this was the basis for our ranking model. Based on this, this was a starting point for the ranking model, and it enabled us to only synthesize high affinity molecules, and instead of synthesizing thousands of molecules like a conventional Protai campaign, we only synthesized about 150 compounds, and in 9 months we got for a very strong and selective compound, and this is our current lead. We can see that we have much stronger effects compared to Pfizer. Pfizer here is in red, and ours are the green and blue compounds. So we have deeper, much deeper results than Pfizer, but also very high selectivity of KAT6A over the entire proteome, and specifically KAT6B, which is a driver of selectivity, and Pfizer's drug is non-selective to KAT6B. And this enables also, on top of the deeper potency, also very strong or better therapeutic index based on less toxicities on the myelosuppression and heme toxicities. We tested this across the board in many different cancers, and here you can appreciate that in green we surpassed Pfizer's drug across many different cancers, from breast cancer through lung cancer, ovarian cancer, prostate cancer, and this is not only working in a specific model, so we have a very strong hypothesis across very different models. And also we have a very strong combination theory where, because we're less toxic, we have better combinability with the current first line of therapy, CDK4/6, AKT inhibitor, PI3 kinase, and so on. And here you can see that there's very strong synergy between our drug and the first line of therapy drugs that are currently treating the ER-positive breast cancer space. How you can take advantage of this capability? So we have two ways that we're collaborating with companies. One is asset deals. If one or more of the assets in our pipeline is interesting, we are open to collaborate early on, like we did with the Regenimo case. And all of these are high-value assets that we're focusing on in very large indications. The second potential is platform deals. Here we're starting from discovery services all the way to end drug discovery with the top biopharma companies, and we already were validated and working with several biotech and pharma companies, including Teva, Alexis, 2 out of the top 50 companies in the world. And it takes a village, like Mati said, on the ecosystem, but here we generated a very strong tech bio team, combination of very strong AI people together with scientists and ex-pharma chemists. And we also are backed by a top world-renowned scientific advisory board that helps us to make the right choices as well as connect to the right pharma companies. And with that, I'll finish the conversation and I'm open to any questions. Thank you.
Thank you, Eran. Our final speaker today is Amir Farhi, Chief Commercial Officer of Scopio Labs. Scopio Labs has developed an AI-powered full-field digital cell morphology platform that's transforming diagnostics and laboratory workflows. Amir, the floor is yours.
Thank you, John. Let me share my screen. Okay, give me a thumbs up, John, if you see my screen. Okay. Hello, everybody. Thank you for coordinating this and inviting us to this great session. My name is Amir Farhi. I'm Scopio Labs Chief Commercial Officer. And unlike my colleagues here who presented before me, we are in a slightly different space. Scopio is a medical device in the space of hematology, blood tests. We are a hardware and software solution, AI powered, and I'll show you what it's all about. And thank you again for having me. My contact information is here. Happy to answer any questions during or after my presentation or anytime after. So, for those of you who are not really familiar with the lab diagnostics and tests, blood test, CBC, complete blood counts, these are the most common lab tests in the world. Every one of us has gone through those probably quite a few times in our lives. And the number of these blood tests is expected to double over the next decade. Just in the US, it's expected to surpass 1 billion tests a year. And this is a trend that is happening globally in Korea and in other developed countries as well. The problem is that while this demand for test is on the rise, the expertise, people that know how to analyze blood test results is not on the rise. It's actually on the contrary. It's on the decline. There are many open positions in the space of lab diagnostics. People are not going into this. While most of the blood tests, the complete blood count is done automatically, there's a portion that is done manually or semi-manually and does require expert review. And this is where the challenge is growing because not a lot of people know and are able to handle this growing demand. So we know, we see, we hear it from all hematology labs around the world on how problematic it is for them to address this challenge. Now, just at a high level overview about blood tests, complete blood count, the blood test I mentioned earlier, most of it is done by CBC analyzer, and that's done automatically. However, when there are any abnormalities, and it's on average anywhere between 5 and 15%. It all depends on what lab, what the population is. Like globally, it's somewhere between 5 and 15% of blood tests do require something called peripheral blood smear review. Somebody needs to look at the blood under the microscope. And although that's a relatively small portion of the tests, because it's manually, it does require quite a lot of the workload, costs, and resources. And as I mentioned earlier, this is something it's a dying breed, the expertise. Solutions out there in the market, there are some semi-automated solutions that have been out there for probably 20, 30 years that try to help the labs, but they're all still I would call them semi-automatic. They still require expert review, a human to review, to look, to sign off, and they always require and are tied to a microscope. So whenever there's a peripheral blood smear review done, even though it's done digitally or semi-digitally, you're always going to find in the lab a microscope right next to it where someone can review the sample. Again, blood morphology is something that is also, it's a lot call it more of an art than a science. It's classifying different blood cells, white blood cells, red blood cells, platelets. And again, it's a dying breed, and demand is on the rise. Before I jump into what Scopio does and how we solve the problem and our breakthrough technology, just a little bit about who we are as a company. We've been around for about 10 years, a little more, 11 years now. We actually started in the vet space, in veterinarian, before we got our FDA and CE and other regulatory approvals. We have, at this point, hundreds of labs around the world using our technology. And as of now, we've partnered with Beckman Coulter and Siemens Healthineers as global distributors for Scopio, but we also work directly. We have a small direct sales force and marketing and customer success, as well as small local regional partners. As I mentioned earlier, Scopio is a combination of hardware and software. So we have our imaging devices, which I'll talk to in a second. We are able to generate digital images of blood and blood type, so bone marrow, using our scanners. And then we have applications on top of that that help the reviewer do the analysis. Let's talk a little bit about what makes Scopio really unique. So our core technology is what we call the full field digital cell morphology. And what's unique about that is that in order to look at blood and blood types, to really do the proper analysis, you need 100x magnification. Even under a microscope, you need 100x magnifications. And technology out there, in order to digitize at 100x magnification, there's never, until Scopio, nobody was able to crack a solution that doesn't have this compromise between the field of view of how much we're capturing when we're taking the image of the blood, and the quality of the image. I mean, 100x, full resolution. When you look into pathology solutions, many of them require only 20x or 40x magnification, and you have many scanners out there, imaging devices that deal with pathology images at their required resolution. However, to look into blood, you need 100x magnifications. Now, traditional solutions such as Cellavision, who's been the market leader for the past 2 decades or so, Mindrays now, actually there's a South Korean company that you may be familiar with called UIMD, also use what's called single cell imaging. So they take single cell images of the cells at 100x magnification. The problem with that is that you get just images of the single cells, not the full area of the blood. Hence, it requires always dependency on a microscope being right next to the blood. Scopio is the only one that, by using computational photography and very powerful processors, that we're able to generate 100x equivalent images of the clinical relevant area. We've got two imaging devices or scanners, if you will. It all depends on how much the throughput and the volume needed by the labs. We have the smaller scanner that does 3 slides at a time, and the big one that does 30 slides. But they pretty much do the same thing. It's just at a different volume, different capacity. And our applications on top of it for peripheral blood smear review and bone marrow analysis are available for both of these imaging devices. Now, the whole concept of Scopio and the full field of technology is that think of it like a Google Maps experience. You see everything. You can zoom in, zoom out, pan around. You can annotate. You can do everything digitally. And that is really what makes a huge difference in the world of hematology, because no longer you have the dependency on the microscope. No longer do you have to have the reviewers sit next to the slide. You don't have to ship slides to the morphologist or to the hematologist, and not the other way around. You could do the analysis, the review from anywhere, because Scopio generates the image digitally instead of looking under the microscope. So we've got, as I mentioned, we've got cleared in the market. We've got both FDA and NCE for the peripheral blood smear review and our bone marrow application review. Peripheral blood smear is also cleared in South Korea, as well as, I believe, in Singapore, Australia, and a few other countries. BMA is in the US and Europe. I'm not sure about regional acceptance, but based on demand, we can always add more. You get the digital image for either the peripheral blood smear at 100x magnification, as well as bone marrow. Scopio is the only one in the market. We were also the first one to get the FDA de novo clearance on bone marrow aspirate application, and the market loves it. I speak to a lot of hematopathologists that just love the digital experience with bone marrow. So first you get the digital image. Again, instead of looking under the microscope, you get a digital image at the 100x. On top of it, Scopio has the AI decision support. So it helps whether it's the lab technician, the hematologist, the hematopathologist. It does the classifications of the different blood cells, the white blood cells, red blood cells, platelets, and so forth. It's still a decision support. It's still an expert needs to review it, but they don't need to be physically next to the slide. You could collaborate with others. You could work from anywhere, and Scopio does a lot of the work for you. 96% of cells, based on studies that we publish, are classified automatically by Scopio. Again, pre-classified, somebody still needs to sign off on it, but it really streamlines the process. As I mentioned, for the peripheral blood smear, it's both white blood cells, red blood cells, and platelets. We also have the bone marrow. Again, one of a kind, a process that is a lot more complicated to do a differential. It's called the analysis of a bone marrow aspirate. You need to classify based on the guidelines, 500 cells. Scopio does it a lot faster for you, for the expert. They can just review, reclassify, add notes, sign off, and off it goes to the LIS, the Laboratory Information System. Now, the thing that, because Scopio was really able to digitize the image, now the workflow of reviewing peripheral blood smear and/or bone marrow can be done fully digital. And as such, workflow can change. You don't have to do those things physically in the lab. You could use remote expertise. You could do workload balancing between multiple labs. Again, no longer the need to send slides to experts or the experts to the slides. You're switching a traditional, very cumbersome and error-prone process on a microscope that is done manually, counting cells manually, to doing something that is now done all digitally from anywhere. These are some quotes from some of our customers from different labs around the globe. Studies we've published and quotes we got from our customers that it's faster, 60% review time faster, and almost 60% reduction in the turnaround time for that. Now, this is what we have currently in the market, the peripheral blood smear and in bone marrow aspirates. What we're coming up next, and it's now in advanced clinical trials, we do, hopefully, fingers crossed, we're going to get the FDA and CE clearance by the end of the year. Can't promise it, but it looks very promising. It's the next generation called complete blood morphology. And on a personal note, that's the reason why I joined Scopio 3 years ago. If I mentioned before the peripheral blood smear review, uh, the routine peripheral blood smear review, even with Scopio, is still requires manual or an expert review to review and sign off on it. It stops the process. Somebody needs to look at the blood, do the analysis. Again, even if it's Scopio, they need to sign off manually. And when you look at the cost associated with the peripheral blood smear review, it's very costly. In fact, we've commissioned a third-party study that was done in the US, and on average, it costs labs, and many labs don't even know how much it costs them. It costs them close to $16 per peripheral blood smear review between the lab technician, expert review, going to the microscope, all of that. Very costly process, and also a process that is not standardized. You'll see that different people sign off different things on a peripheral blood smear review, and sometimes you'll see the same person giving a different review and analysis because it's amorphic cells, and a lot of times it's difficult to determine what's what. What Scopio is coming up with now is our CBM analyzer, the complete blood morphology analyzer, autonomous analyzer that will sign off automatically on this, on the peripheral blood smear. And the reason why we're able to do that is, again, because we're able to digitize the entire, peripheral blood smear slide, or at least the entire clinical area. And instead of looking just at 100 cells, white blood cells, we look at 1,000 blood cells and 10,000 RBCs. So the guidelines for reviewing peripheral blood smear currently in the market, according to the ICSH guidelines, are based on human limitations. Scopio broke that glass ceilings. I'm sorry. We don't have this human limitation anymore. So from morphology, we're going to a morphometry. We're looking at a lot more cells. And from a review perspective, it's, from a statistical perspective, it's likely going to be more accurate just because you're looking at a lot more cells. The result of that is going to be a lot more, reduced cost in peripheral blood smear review, staffing optimization, standardization of the process. No longer you have this different people calling different things different ways, and you're really gearing up your lab towards growth and efficiency. In a recent study we've done with 2,500 peripheral blood smear, Scopio CBM analyzer signed off automatically on 93% of the cases. No human intervention. This is truly bringing total automation to the hematology labs. So just to summarize, Scopio, we are the full field digital cell morphology. We've got the imaging devices, and on top of that, the peripheral blood smear, bone marrow, and coming soon, the complete blood morphology. Thank you very much. Again, open to question. Thank you, John, Tamar, and happy to answer any questions you may have. Thank you.
Thank you very much, Amir. Before we wrap up, we have one question in our Q&A chat box, and I believe that question is for you, Malti. Malti, if you can please answer that if you can, otherwise I'll wrap up.
Yeah, sure. Can you let me see this. What's the question here? I can't really see it.
Okay.
Yeah, what specific collaboration model do you expect in Korea? Okay, so maybe I can actually give an example that I discussed before, but I'll try to kind of give it specifically. You can see here the slide that I pulled up, which is the example that that one of our startups called Profit is doing with Dongbang FTL. And Dongbang is a great Korean drug discovery company in the small molecule space, and they were looking to be able to actually become a little bit more AI driven on the innovative side. And what Profit is doing is using their core technology to actually come up with new hits in a very rapid manner. They just completed their first POC. We don't get to have the results to be able to share, but based upon Profit's track record, I'm sure it was very successful. And to be able to develop a molecule for Dongbang. Now, the way that this happened is that Dongbang ultimately owns the IP rights to the molecule, and Profit is receiving a service fee as well as also wet lab validation in their initial stages where they're just 1.5 years old. So it's really a win-win in this aspect. They jointly applied to a government grant that you can see up here from CoReal, and it's a joint fund. Eran also mentioned this between Korea and Israel. And so they're able to each get funding from the various government. And we know that as much as the Israeli Innovation Authority funds Israeli companies, Korea even does it much more intensively. And so each company was able to receive a small grant. And I could see replicating these type of very complementary collaborations at a large scale. So each one of the Aion Labs startups could do these type of projects with good Korean companies that are looking to develop more artificial intelligence and technological capabilities. And you could do the same with CytoReason, you could do the same with Protai, you could do the same with Scopio, and you could do the same with any other Israeli startup. So if anyone out there, whether you're in Korea or in other Asian countries that are on the line now, if any of you are interested in AI capabilities or technological capabilities in drug discovery and development, you can reach out to me. And if we don't have an offering for you at Aion Labs, we're happy to connect you with other ecosystem players. You could reach out to the embassy. You have a great team here from the embassy team that really knows how to reach out to us. This was born actually because Dongbang was looking for these capabilities and reached out to us based upon a LinkedIn post that I put up there about Aion Labs. They asked us, but they were very specific about what their needs were, reached out, and we connected them with Profit, and this is how this was able to happen. So hopefully that answered the question.
Thank you very much, Mati, for that. And thank you to all the speakers for sharing such interesting, valuable insight with us today. I would also like to thank everybody who joined us. We really appreciate you taking the time to be with us. We hope today's webinar has been given a better understanding of Israel's pharmaceutical and healthcare ecosystem and sparked new ideas for future collaboration. Before we close, we would like to let you know that a short survey will appear after the webinar. Also, the survey will be emailed to all of you. If you're interested in having one-to-one B2B meetings with any of today's presenting companies, please take a few moments to complete the survey. Your response will help us understand your interest and arrange potential business meetings with the companies. Well, thank you again for joining us, and have a great afternoon in Korea and great morning in Israel. So goodbye, everybody.
Thank you. Goodbye.
Bye. Thank you.
Bye. Thank you, everyone.